Hey, here we are. Let's see your faces real quick. Quickly, my friends. Bring your faces up. Simmons. Simmons wins. She's first. Nishi comes in second. Esha comes in third, but she doesn't need the bonus points. She's already doing well in this class. Stephon McCain, still nothing. Tyler Montano, how you doing? I keep hearing notices that you're not well. I hope you're okay. Vanessa? Where's Bimalek? Abimelech. There she is. The jungle, the mighty jungle. Abimelech sleeps tonight. An African song.
I really had difficulty with today's quiz.
Oh, you took it already. All right, if you haven't taken today's quiz, go ahead and do that. It's open until 7 minutes after, so you have 5 minutes. Actually, it won't take nearly that long.
Yes, it's until 2 o'clock. You can build the bear. Where? School. Sure, we need to go now because I also got to spell to my professor. All right.
Vanessa, do your quiz.
I already did it.
Already did it.
Yeah.
Does anybody need more time with this quiz? It's a difficult one.
This quiz is very interesting.
Yeah. It's a hard one. Okay, if you're coming in late, you only have 4 more minutes to do the quiz. Hurry up. Hurry up. Oh, Vanessa's got her puppy with her. Hello, puppy. What's the puppy's name?
It's Rocky's puppy. Her name is Maggie.
Maggie. That's a great name.
Hey, Maggie.
Hey, Maggie. Arf, arf. Arf, arf. Okay, 1 more minute for that quiz, and then we're going to start the learning. The learning shall begin. I'll do the teaching. You'll do the learning today. Just to keep that straight. Just for lols. I hate that term. Let's just do it for lols. Time counting down on the quiz. 5, 4, 3, 2, 1. I'm just making those numbers up. I don't know. Did you give us the results of the poll about the next test? I did. The next test will be on Monday, April 5th. It will cover, before anybody asks, it'll cover everything we did in class since the last test. I always get that question, what's going to be on the test? What do you think? Everything we talked about. I'm talking about this for a reason, people. Not you people. You're smart.
Wait, is the quiz supposed to be like this?
Is what?
Is the quiz supposed to be like this?
It's the quiz. It's the quiz.
Honestly, this is probably the hardest quiz I've ever taken in my life.
Yeah. Well, hopefully you didn't put absent. Yeah, I didn't have time to make a quiz today. I was putting my lecture together until like 10 minutes to 1. I was like, oh no, I didn't do a quiz yet. And I couldn't think up a question quick enough. So I thought, we'll just give you credit for showing up today because we've got 27 out of 35 people. So not everybody got the bonus points there. Although I swear to God, I'll get an email. Oh, can I take that quiz? I was a little late. No, you may not. Okay, let's rock on here.
I have a question before.
Question?
Can we do that?
Sure. Question. Nishi.
So, for lecture 7, it says, like in the notes, that genes of the cells in the presomatic mesoderm cycle at 90 minutes per cycle, and then genes get turned on by FGF, and then they turn themselves on. But like later, when we go towards the end, it's like Wnt turns on the HOX-A gene. So, like, is it Wnt or FGF that you're expecting? I mean,
It's different genes we're talking about. So Wnt is turning on. FGF is turning on Wnt. Wnt is turning on Notch. Notch is turning on Ephrin A2. Those are the genes that are cycling for the clock. And the reason for that is when that Ephrin A2 comes up, it becomes a somite. So it keeps cycling through that till you get past the FGF gradient. So the Wnt HOX thing, that's like on its own clock. It's going at a different separate thing.
all right. Okay. Thank you.
Sure. Who got the best video of the HOX genes? No one did. No one got what I was looking for. I think I may have to make a video myself of it because no one really came up with what I was hoping for. So sorry, guys.
They had no good videos on the 5 minutes.
Yeah, everything was like,
Yeah, the videos are like half an hour and stuff.
yeah, or else they were about Drosophila or invertebrates. And it's like, we're not even covering that. So just give me the points. Okay, Yusuf, Mr. Point Grubber, I beg you. Begging sometimes helps. Okay, so let's rock on. So we're still, now we're going to be patterning within one somite, front to back and left and right, or not left and right, dorsal ventral. Left and right too, I guess. Sure. Sharing is caring. Sharon is Karen. Karen is Sharon. Okay. So quick review of what we talked about in the last class, since we typically have more time than we need here. So I think Notch was the class before, but when you get upregulation of Notch, that's what causes those somites to separate. Not Notch signaling does it. So when you get past the determination wavefront and you have Notch signaling going on, you get Ephrin A2 and boom, you get a somite. But we'll see that even at this point, you've got Notch signaling here, but no Notch signaling here. So we've got some anterior, posterior patterning within a somite based on Notch signaling, but that's what we'll talk about today. And then here we're talking about how your pre-Semitic mesoderm is already patterned. So those cells already have certain genes turned on as the node is going forward. And the reason for that is here's our determination wavefront. This is somite formation. This is your clock. So this clock determines how many somites the organism is going to have, because those cells sitting in the presynaptic mesoderm are putting out FGF. FGF will make Wnt. Wnt will make Notch and axon, which turns Wnt off.
Did you say presynaptic?
Presynaptic. Oh, God. Presemitic. Presomitic, not pre-Semitic. Pre-Semitic is before Jews and Arabs. So this is post, well, actually it's pre-Semitic too, because this was happening before there were humans even. I'm on a tangent here. Let's get back to work. All right, so in that pre-Somitic mesoderm between the node and where the somites are forming, you've got these genes cycling through every 90 minutes. Now the movement of a cell from one end of the pre-Semitic mesoderm to the other might take a day. So during that period, you've got the cycling going on every 90 minutes. So FGF produces Wnt. Wnt produces Notch and shuts itself off. Notch produces Hairy and Ephrin 4, and then shuts itself off. And then eventually this will get broken down and everything gets broken down. Axon gets broken down. Lunatic fringe gets broken down. Now FGF can make Wnt again. Wnt makes Notch again. Notch makes this. Blah, 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 blah. Keeps doing this until that particular cell gets past the determination wavefront, because that determination wavefront is based on the concentration of FGF. So FGF drops at that point because you've got RA coming from the other side, and that shuts this whole thing off. And so that last time that FRNA comes up, boom, you get a somite. Okay, that's our clock. And this is again just showing, oh, so now this is showing how that pre-Semitic mesoderm is already patterned, even though it has not become a somite yet. It already knows, those cells already know what somites they're going to be in. And that's because of the Hox genes that are also kind of cycling through on their own time frame. So first the Hox1 genes will come on, and whatever somites get made while that one is on will become the top somite, which will be hindbrain, in the area around the hindbrain. And then 2 and 3 are also kind of hindbrain. So those somites, these genes are on when those somites are being made, all the way till you get to the other end of the spine. And now all the genes are on, and they all are turned on in those last couple somites. And that makes that posterior. And you've got combinations of genes that are important for each somite and patterning each somite anterior to posterior. And again, these, the ones come on first and they stay on the whole time, although their highest expression level is right at the beginning. And then the next ones come on, and the next ones come on, and the next ones, and the next ones, and the next ones. And it's cumulative. You know, at this point, you've got all the genes on, but only the last somite has this one on. And the reason why they're cycling is because oh, this is cool. I like this. This is just PowerPoint animation. It's not anything special. So you can see when the first somite comes out, when 1 is on, so it's got that pattern into it. The next one's got 1 and 2 into it. Here comes 3. And skip to the end because I got tired of doing it. And there you are. Okay. So the reason these things are cycling is because your Wnt turns on the first ones, and the first ones then turn on another signal, CDX2, that turns on the later ones. And then when you get all the way to the end, Hox18 shuts off the earlier ones, and then they all shut off after that. So it's sequentially programmed into the DNA. Okay, so now we're up to today. First thing to talk about real quick. So today we're talking about anterior, posterior patterning of individual somites, and also dorsal ventral patterning of the somites, and left-right patterning of the somites. Although we'll get more into detail with left-right later. But anyway, first and foremost, anterior, posterior. So if you remember back to that previous slide, I said that you've got notch signaling at kind of the posterior end of the somite and no notch signaling at the anterior side of the somite. So that's what this is showing. This is a somite, red to white. So the red is the expression of MSP2. And if you remember from that slide, MSP2 inhibits notch signaling. So we've got no notch signaling here. On the other side of that somite, though, let me blow this up. So here's anterior and posterior side of one somite, and we've got no notch there, we've got notch here. Here's that same somite, but now we're looking for notch, and you can see delta shows up here. It doesn't show up there. So delta is the ligand for the notch receptor. Okay, so MSP blocks delta notch. Delta notch produce MSP, and so you end up with this anterior posterior patterning within one somite. Not that big a deal, but there it is. All right, more important and more complicated is what's going to happen from the anterior to the posterior side. Okay, so here we are. This is post-neurulation. So you can see your neural tube is closed. You've got the nodal cord under the neural tube, and you've got epidermis closed on top of it. Yusuf Okind, what cells are here? You can't see them right now, but later we'll talk about them. What cells are on top of the nodal cord and migrate away? Yusuf?
Um,
Let me give
trying
you a
to
hint.
think.
They're at the crest of the neural tube.
Is it? It's not the. It's not the. Is it mesoendodermal cells or no? No,
It's at the
I guess
crest
not then.
of the neural tube. I can't read the chat while I'm in this form. Nishi, tell him. Tell him or I'll take 3 bonus points away. Nishi?
I put it up in the chat. I mean, instantly.
Oh, you did?
Neural crest cells.
Neural crest. That's what my hint was. I said it's at the crest of the neural tube, neural crest cells. So anyway, that's just a little review from chapter 3, but we'll get back to it by the end of this chapter, I think in our next lecture. But anyway, when this neural tube closes, some of these cells become neural crest cells and they become mesenchymal and crawl away. And that's what's going to form the nerves that innervate that somite area. Okay, now here's your somite, right? Up to now, we've been just saying it's a block of tissue. And the reason we've got a somite here is we went passive determination wavefront and went through the mesenchymal to epithelial transition, right? So they went from crawling cells to stationary cells. And now they're a somite. But now, as they're sitting here, they're getting hit with hormones from different directions. So we're getting we've got 3 different gradients of hormones going on here. We've got Wnt coming from the top, we've got BMP coming from over here, and we've got sonic hedgehog coming from down here up. So those hormones we'll talk about in a minute and how they work. First, let's see what happens to these cells because of these gradients. So here's your somite. Again, it starts out as just one solid block of generic somite cells, but pretty quickly, they start to differentiate into different types of cells. So on top, you're going to get dermomyotome. So the dermomyotome is going to be dermo, skin, myo, muscle. So right now it's pre-dermo, pre-myo. And the reason for that is it upregulates the transcription factor PAX1, I believe. PAX1, where am I? Make sure I don't say that wrong. PAX3, sorry. That's PAX3 makes this into dermomyotome. And then down here, the blue part is the sclerotome. Sclerotome will become bone or spinal column, basically. Those cells will crawl around the neural tube and form the vertebrae. So and they are sclerotome because they've upregulated the transcription factor PAX1. So PAX3 makes dermomyotome, PAX1 makes this sclerotome. Okay. Now, in terms of this dermomyotome, let's see what our next slide says. This dermomyotome, well, all of them now that they're getting hit with all these hormones are going to do the opposite transition from what they just did. They're going to go from epithelial back to mesenchymal cells. So before they did the MET transition, now they're doing the EMT transition. So that block of tissue now is going to fall apart and they're all crawling off in different directions based on the concentrations of the hormone gradients that they're seeing. And this dermomyotome here, this dermomyotome will then differentiate into dermatome and myotome. Okay, so those 2 types of cells are going to separate. The dermis is the skin, okay? Underneath the epidermis is the dermis. I'll show you a picture of that in a minute just so you can kind of see the pants, understand what that means. But it's going to form the skin layer that's underneath the dermis. Myotome then is going to become muscle. So this transcription factor comes up, MyoD. So dermatome doesn't have this. They both have PAX3, but myotome gets MyoD. And MyoD is going to make these things migratory and going to make them muscle. Now at this stage myotome, you've got two different types of myotome. You've got medial myotome, that's going to stay in the middle. So that will be back muscles and everything going down the back. And then you've got lateral myotomes, that's going to go around the body to make the abdomen. And then also limbs will come out of those. If that particular somite has got a limb programmed into it, it will come out there. And then our sclerotome then, the most ventral part, it's got PAX1. It's going to surround the notochord to form the vertebrae and the ribs. The ribs. Do you ever notice how Southern people put extra syllables and things? Ribs. I love my ribs. Okay. So here we are. I'm going to show you a video in a minute where you can see this a lot better than this particular picture. I'm not crazy about this picture, but you've got some of these dermomyotomes becoming medial myotome, and they're going to crawl all around in this area and become back muscle. And then you've got the outer layer, which is going to be dermatome. It'll be right underneath the epidermis. Then you've got other ones, lateral ones, are going to crawl sideways and go around the body and also form limbs and things like that. And then here's your sclerotome that's going to go around from either side of this and make make your vertebrae and ribs. Okay, dermis, axial and back muscles, abdominal and limb muscles, vertebrae and ribs.
I'm sorry, but all of these things, I mean, like every part of this undergoes EMT or like?
Yes, yes.
Okay.
All four of these different types of cells undergo EMT, and then they start crawling off and become what they are. Okay? So kind of to put that all in a scheme, they're mesenchymal when they're in the pre-Semitic stage. They go past that, the determination wavefront, and they go through MET and they stop. And then they get hit with a whole bunch of different chemicals, and then they let go and start crawling again. Okay, so they go through the transition twice, and they'll go back to, they'll go through the other transition again, right, and become stationary again eventually. Okay?
Yeah, thank you.
Okay, so this is what I told you I was going to show you, the dermis. So epidermis is ectodermal, dermis comes from the mesoderm. And this purple line here is your basement membrane, or what else is it called? Basement membrane. Oh, I can't remember what it's called, but basically it's a really thick layer of protein. So there's a definite break between the epidermis and the dermis, and cells of the epidermis can't come through this way. So that I'm just throwing that out there because when we talk about if you take my cancer class next fall, we'll talk about how most cancers are epidermal, and they're not considered cancer until they go across that basement membrane. So cells will start to crawl and move around, but that basement membrane helps to protect the rest of the body from whatever crazy stuff's going on in the epidermis. Okay, not that important. All right, here's the video I wanted to show you. So let's do this. This is about 8 minutes, and he's really good. So let's do this. Oops.
Segments of somites, and there's the cervical somites and thoracic and lumbar somites, as well as sacral somites. And then we have at the tail end some of these coccygeal and then more cranially pre and post votive somites. Considered altogether yellow and purple, there's 40 to 42 somites. We're going to focus on the somites highlighted in those dotted lines. So the 8 cervical, 12 thoracic, 5 lumbar, and 5 sacral somites. We're going to take one of those segments, take a cross section through it, and ta-da! There we have this cross section through a somite, and one somite highlighted in yellow. In this picture, there's the neural tube, who becomes neural crest cells, become the dorsal root ganglion, and simply for orientation, there's the gut tube with the associated solomic cavity, and all on the other side, shing, there we've got the soma. So let's talk about the dermatome, myotome, and sclerotomal components, those three elements that comprise these condensations of periaxial mesoderm that we know as the somites. So first in pink, we have the dermatome. These dermatomal cells are more superficial in this somite, and they're going to migrate out and become the dermis of the skin of the back. The overlying ectoderm is derived, it becomes the epithelium. The dermatome becomes the skin of the back. As you go forward more, you get this ring, as you can see in this picture in pink. The body wall and the skin of the limbs comes really from the somatic part of the lateral plate mesoderm, but it's from the same segment, so the principle's the same. And there we have in pink this ring of skin, and in yellow there is our spinal nerves. And so the dorsal root is primarily sends off branches to provide sensory innervation of this, the dermis of the dorsum of the back. And then this ventral ring, of course, gives rise to a lateral branch that provides sensory innervation of the lateral part of this band of dermis, and then it terminates as an anterior cutaneous branch, which sends sensory information from the front. And therefore, we see this entire band in pink. It's innervated by all the branches bilaterally of dorsal and ventral rami. It forms this ring of innervation of the skin. Hence the clinical definition of dermatome, an area of skin supplied by a single spinal nerve level. This segmental happens 8 times in cervical, 12 thoracic, and so forth. Myotome is the region highlighted in purple and red because myotome has 2 parts. The epimere, which are these cells that migrate more dorsally to the epiphyseal pond, the top part of this developing embryo, and these cells are going to develop into the erector spinae muscles. Whereas the hypomere in red, these cells migrate more ventrolaterally and they form the body wall muscles,
So this is what I was calling this medial and lateral. So the medial then makes your back muscles, and the lateral then makes the whole body cavity, surrounding the body cavity, rectus abdominis, and all those muscles.
below on the bottom of this developing embryo. But in the cervical and lumbosacral regions, becomes muscles of the limbs. The yellow arrow shows these, the epimere becomes these epiaxial muscles. And the dorsal ramus, that dorsal branch, will innervate not only the skin of the back, but also these epiaxial muscles like erector spinae. In the hypomere, these cells form the hypaxial muscles, which is 4 layers of lateral body wall, subvertebral muscles, and the ventral strap muscles. And as I mentioned before, in the cervical and lumbosacral region, hypomere components of the myotomes become muscles of the ribs. The ventral ramus branch will innervate all muscles of the body wall and the upper and the lower limb muscles. Finally, in green, there we have the sclerotome. These sclerotomal cells will migrate and surround this branch.
Let me just back up for a second. I want to explain something else that's going to be important to you in the future. Probably not in this exam, but somewhere in the future. If you look at this structure here, you've got muscle, and you've got bone, and then you've got muscle. This is your T-bone steak right here, okay? The butcher cuts through this way, and you got there. So you've got this part, and you've got this part. Now this part is called the tenderloin. This is the most soft part of the steak, okay? And sometimes you can get beef tenderloin, which is just that long section right out of that underneath the spine part. Softest, easiest part to eat.
Muscles of the body wall,
just saying.
The upper and the lower limb muscles. Finally, in green, there we have the sclerotome. These sclerotomal cells will migrate and surround this developing neural tube, the spinal cord. So there we've got the neural tube surrounded by these bony segmental elements we know as vertebrae with these costal components. These are segmental from top to bottom, and so they protect the spinal cord, but there's spaces in between for the spinal nerves to exit at each segmental level, and also allows for some flexibility. So here we have that picture now highlighted with the dermatome, epiaxial, hypaxial muscles, and the sclerotome component. Looking at this lateral view again, and we take one segment and shing, we blow it up, and there we have a segment. All those components, vertebral, spinal cord, spinal nerve levels with dermatomes and myotomes. But how about this level? Same thing at that level. There is a vertebral level, spinal cord level, associated spinal nerve with dermatome and myotome. Oh, what about this level? And the same.
Swing.
The key to this is
Swing.
every single segmental level, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral levels, there is going to be a vertebral element. There's the vertebral element. There's going to be a spinal cord element. There's going to be a spinal nerve element, dermatome, and myotome. Those 3 themes we're going to see over and over again now as we look at the neurological levels of each of these regions.
And that's it. So you can go back and watch that again, but what I really like about that is the way he divides the somite into 4 different sections. So you can see each of the 4 sections going. On our on the figure from our book, the dermal myotome looks like one section, and it's like, where are all these cells coming from? But I really like the way he does it. Anyway, you can go look at that again if you want. Now, question number 2 becomes why? Why in the name of God does this happen? Okay. Okay, so there are your migrating cells, blah, 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 the dermis, this guy, and here we are. All right, so here are your gradients of hormones that are patterning this. Up on the dorsal side, you've got Wnt. So you've got Wnt being produced by both the epidermis and the top of the neural tube. So that's producing a gradient from here down. So ventral is low Wnt, dorsal is high Wnt. Then you've got over here BMP. Where would we be without BMP? What an important hormone. It's being released over here laterally and coming in this direction. So now you've got kind of medial lateral concentration gradient there. And that's your another gradient. And then you've got sonic hedgehog, which is another hormone, and it's being released by the notochord and the neurotube on the bottom, and it's going up. So we've got these 3 different gradients: sonic hedgehog from the bottom up, Wnt from the top down, and BMP from this side over. So that's how that somite is going to get turned into 4 different pluripotent tissues, because each one is going to be exposed to a different concentration of each thing there. Okay, comments? Nishi, how does this sonic hedgehog mechanism work? You don't know, do you?
I have no idea.
Finally, something I found something Nishi doesn't know. I'm teasing you. I'm teasing you. Okay, so this is how sonic hedgehog works. It's kind of a strange, in my opinion, kind of a weird mechanism too. So you've got this. Oh, I should have the words up here. Let me put them up. You've got a number of different proteins here. Come here. Come here. Hang on. There it is. Again, we're on Drosophila naming convention, so they're kind of weird. You've got the receptor for sonic hedgehog, which is called patched. Oops. Now, if it wasn't patched, what might it be? Smoothened. Patched and smoothened, smoothened. And then you've got this signaling molecule GLI. And there are a bunch of other molecules, things going on here. There are kinases phosphorylating GLI, and there's ubiquitination and proteolysis and all this stuff going on. Leaving out all those crazy steps, we'll just look at the basics of this hedgehog mechanism. Okay, so when you don't have when you don't have any hedgehog around. So basically the mechanism is turned off. What happens is patch blocks smoothened from going into the membrane. So this membrane has got these primary cilia that stick out of it. Okay, so like any other cilia, except they're not used for movement, they're used for signaling. And we'll talk about cilia when we get into left-right stuff too. But anyway, you've got these primary cilia and glia is up in there. Now, because smoothened can't get to it, patched is holding it back, it gets proteolytically cleaved into a portion of that gli protein, which is called gli3. It's a repressor. So this will go to all the genes that that hedgehog mechanism will turn on and repress them all. So all these genes are held off by this gli being broken down into a repressor. Now, when hedgehog is around, sonic hedgehog then binds to patched, and now patched is no longer blocking smoothened. So smoothened with that whole vesicle fuses into the membrane, smoothened goes up to the primary cilium, and then it causes gli to be proteolized in a different way. It gets cut, but now there's more different amino acids present. So now this is an activator. So it's going to go to the genes, kick off that gli repressor, and turn some genes on. Okay, should probably put that here. Gene activation.
I'm sorry, I couldn't follow. What gene are we activating? Like, what's this pathway doing?
Okay, let me, I'll back up and do the whole thing again. Let me just do this. Bum, 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 bum. Okay, so let's back all the way back up to the gradient figure and then go through this whole thing from that direction. Okay, so you've got Sonic Hedgehog being released from down here up here. So up here, where Sonic Hedgehog is low or totally absent, you're producing this gli repressor from the gli protein. And that will turn off any gene that Sonic Hedgehog would turn on. Down here, where Sonic Hedgehog is high, you've got it binding to patched, which then no longer blocks smoothened. Smoothened goes into here. Now it gets cut into a different piece, and that activates a set of genes. Okay, so basically, whether it gets cut into the repressor or the activator determines which genes get turned on. So if we go back here, you can imagine certain genes are getting turned on here, and again, it's a gradient effect. Different genes getting turned on here, and different genes being repressed up here. Wnt, some genes here, different genes here, different genes here. Same thing with BNP. And you can imagine for any position a cell is in, north, south, east, west, front, back, right? It's going to find a different concentration of these three things. And going all the way back to the last example, when you've got a gradient, you can have differential effects all along that gradient. So now you've got a three-dimensional, three different hormone gradient of things going on here. Plus you're notched back and forth, right? The first thing I said was you've got notched at the posterior end and no notch at the anterior end. So a lot of different signaling going in different directions here, and that's what's giving you your axes there. Okay. Questions, comments? Shall I go over it all again? I got 10 more minutes, and that's about what I wanted to cover today. Basal line. Okay. Questions, comments, anything? Okay, so that's what I wanted to cover today. If you want to go, you can go. If you want to stick around for the next 10 minutes, I'll go over that whole schmear again, just to make sure everyone's on top of it. Okay, later, dudes. I will go back to this and start over. Okay, so in terms of patterning our somites, we've got a little anterior posterior patterning going on. Not a lot, not that complicated. You've got at one end of the somite, you've got MSP being produced, and MSP is inhibiting Notched. At the other end, you have Notched and delta. So the receptor and its activator, I guess you would call it, or hormone in the front. So that's a little AP patterning. In terms of left-right patterning and up-down patterning here, so this would be anterior and front, posterior and back, medial lateral, dorsal ventral. So in terms of dorsal ventral, your somite first turns into dermomyotome versus sclerotome. Sclerotome will become bone. Dermomyotome will become skin and muscle, dermomyo. So then they undergo this mesenchymal epithelial to mesenchymal transition. So they start to crawl, and you end up with the myotome then turning into dermatome, which is skin, be right underneath the epidermis. And then you've got medial myotome, which will be back muscles, both inside and outside, I believe, all the axial muscles, the ones on the inside of the spine and the outside of the spine. And then you've got your lateral myotome, which will cover the abdomen and grow limb buds if you've got limbs coming out in that particular somite. And then you've got your sclerotome on the bottom that's going to circle the neural tube and make the vertebrae and ribs. There we go. Vertebrae and ribs. And there's your dermis versus epidermis. I wanted to make that clear. Here's this great video. And here are the gradients, Wnt, Sonic Hedgehog, and BMP, which make all this differentiation happen the way it does. And here's your hedgehog pathway. Patched, blocks smoothened. And this gets broken down into a repressor. With hedgehog around, hedgehog inhibits patched. Smoothen moves up into the primary cilium, and it gets broken down in a different way and becomes an activator. Okay, that, my friends, oh wait, I've got it chat here. Could Cerberus block somites from forming? Cerberus. I think, trying to think now. Cerberus, I think it depends on where we're at in differentiation. If that's a really good question. I think no, it wouldn't stop somites from forming, because somites are forming because of that FGF Wnt. FGF, yeah, it would, yes, because Cerberus blocks Wnt. Am I right? I think it blocks Wnt and BMP. So if we blocked Wnt, then you wouldn't get Notch, and then you wouldn't get F2A, EF2A, or whatever. So you wouldn't get somites. But if you let that somite form, and then you hit it with Cerberus, then you would be blocking the if you let Cerberus form, you let the somite form, then you hit it with Cerberus, I'd be willing to bet it would block this and this. So all you would have would be Sonic Hedgehog. So this would probably all become scleroderm, right? Sclerotome. That's what I would guess. This somite here is being hit by Wnt and BMP, and that's making it dermomyotome. These bottom ones are getting hit mostly with Sonic Hedgehog, so that's making that into sclerotome. So I would guess that if you let it get to the somite stage and then hit with Cerberus, you would block both of these, and this would all become sclerotome. But if you hit it with Wnt back here, okay, if you hit it with Cerberus here, a lot of this has to do with Wnt signaling. So you're knocking Wnt signaling out. They would probably, I'm guessing they would go through their, yeah, I don't know. Because at this point, you need Wnt signaling to get to Notch to get to making somites. So if you knock Wnt out here, you wouldn't get somites. What would you get? Probably just undifferentiated tissue. I don't know. That's a good question.
Also to add on to that, like Cerberus, doesn't it block the Sonic Hedgehog? Like it doesn't block that?
I don't know. I don't think so. I mean, this is the first time we're seeing Sonic Hedgehog, and previously when we saw Cerberus, we said it blocked both BMP and Wnt. But here we're talking about FGF and Wnt and Sonic Hedgehog. I don't know if it blocks Sonic Hedgehog. I wouldn't think so, just because I haven't heard of it before.
Okay.
I don't know, though. Yeah. Kind of, you know, so here's the thing. Here I'm sitting talking about what I think. That's hypothesis generation. So what I've said might not be true, but if I asked a question that, who asked me this question? Tyler, if I asked you that question that Tyler asked, you don't know the answer. I don't know the answer. But hypothesis generation, you know, what does Cerberus do? It blocks this and this. What would happen if you didn't have this and this? Well, I guess it would be this. And as long as your answer is like logical and based on facts, you know, I have to give you 100%, right? Well, that was fun. Everybody had enough for today? Can we say goodbye? Bye. Get a thumbs up. Goodbye.